left, right.
Good morning, everybody. Thank you for being here. I would like also to thank the organizers for giving me the opportunity to speak here today, and also for funding me with the CAMDA Travel Fellowship, which I'm very glad to have received. I am Moreno Zolfo. I am a first-year PhD student at the Laboratory of Computational Metagenomics at the University of Trento. And I would like to talk to you about the strain-level bacterial and viral diversity in the MetaSUB dataset. But first of all, a little bit of introduction about the kind of the stars of the show. And we all know that bacteria are important as they are the most abundant life form on Earth. And they live and thrive in a variety of environments, from animals, including humans, the environment, and even our cities. And one of the reasons why they are so important, one of the reasons why we are so interested in studying them, is because they are part of the so-called human microbiome, which is the collection of all the microorganisms in contact with their human hosts. And lately, there has been a lot of interest about microbes in humans, the microbiome, because of the, well, first of all, because some of them can be pathogens, and also because even commensal and beneficial strains are important in maintaining the human health, and microbes are important even in disease. But we call the same thing with very different names. And indeed, let's take as an example E. coli. We call E. coli the beneficial and commensal strain that usually inhabits our gastrointestinal tract. We call E. coli the strains that populate the cattle and the animals, guts. And we call E. coli bacteria that live and thrive in the environment, in ponds and lakes and even in the soil. And we call all of these very different things E. coli, so the same species, the same name. And when things go bad, you'll certainly remember the German 2011 E. coli outbreak, in which more than 300 people got involved, and some of them eventually even died. And again, this is E. coli, the same name and the same species. So we may want to ask, what is different between these E. coli? What is that makes the E. coli, the good E. coli good and the bad E. coli bad? And that's true for any species that may come to your mind. Any bacteria has enormous differences in its genetic arrangements. And what can it be? Well, it can be the presence of a particular toxin, a particular antimicrobial resistance gene, the particular set of different antibiotic resistance genes, sorry. And we've seen that with the German outbreak case, in which there was the presence of the particular set of things that made E. coli a deadly strain. But it can be also the belonging to a particular subclade, and we've done this very interesting work with Doyle Ward, in which we demonstrated that a particular substrain, subclade of E. coli was responsible for the majority of the cases of necrotizing enterocolitis, which is a dreadful disease that affects newborn especially, and may also lead to death. So we want to look at the genetic diversity of bacteria, and again, I'll take as an example E. coli, because two E. coli can diverge by up to 40% in their genomic content, and they can be indeed very different. And we want to be able to highlight which are the features that make the different members of the same species so interesting to be studied, being that because we are interested in antibiotic resistant genes, or being that because we are just interested in how their genome, what is present in their genome. So to be able to identify the key features that make strains, that characterize strains. And this has been possible for a long time in molecular biology, and also by sequencing cultivated and isolated strains. And a number of very well-established techniques has been developed to do that in the years. But the problem is that these techniques rely on the fact that we have to cultivate microbes and to be able to isolate them. And another key point here is that they are essentially ad hoc based studies. So if you're interested in E. coli, fine, you cultivate your E. coli and you sequence it and you get everything you want to know. But you can't really expand it to the scale of like city microbiomes. You can't really do that for every microbe. So another approach is metagenomics, in which you can concentrate on the entire genetic content, of a sample by looking at the entire community with one big sequencing. And the problems were that so far methods to achieve the same level of resolution of cultivation-based methods was quite daunting. And in the last years, methods to be able to bring this kind of resolution to the metagenomic field appeared. And this is pretty much what we've been trying to do in our lab by bringing together the best of both worlds and to be able to achieve the strain level resolution with a metagenomic-based approach. And I'm presenting here 3 tools that we have been developing in the lab quite recently. So we start from the idea that a strain is a specific concentration, a specific collection of, in this case, for example, SNPs in the core genome. And we are tackling the problem from the genetic point of view by looking at sub-features and specific SNPs into the selected genes. And this is pretty much the approach of Metamlesti, which is specifically targeted to pathogens, or a more in general approach. This is the approach taken by Strainflan. Or we can take it from the genomics point of view and think that a strain is a particular set of different genes, of the presence or absence of different genes compared to the pangenome of the species we are looking at. And this is the approach of Pamflan. And all these tools are open source and available at the web pages of our laboratory with plenty of tutorials and how-tos, so you feel free to download them and try on your samples. I'm focusing a little bit more on Metamlesti because it's the one I personally developed. So Metamlesti works by exploiting a well-established molecular biology-based technique, which is the analysis of a subset of hypervariable loci in the genome of the species of interest. And it takes advantage of a wide number, of a broad number of available information from other studies, which are publicly available into servers, into public servers. And the way Metamlesti types is basically, backup solution. Is basically to look at specific variants of hypervariable loci, and it assigns a unique identifier to each combination of those. And Metamlesti is able to do this from metagenomic data directly without the need for assembly, and it can detect both previously observed strains and totally new strains of the same species. As an example, we applied Metamlesti on a set of E. coli enriched samples coming from different public studies, including the HMP, the German outbreak gut samples from Lohmann and colleagues, and a set of Chinese gut samples from diabetes cohort. And we were able to picture the diversity of E. coli. I stress it again, this is the same E. coli, the same species, but we are seeing the diversity at the strain level. And we were able to look what you're seeing here is a minimum spanning tree, and each node is a strain, is a sequence type, so a kind of a strain, and they are connected if they share up to one of the surveyed loci. And the size scales with the abundance. And we can see we could highlight very well the presence of a non-pathogenic commensal strain of clonal complex stem enriched in the Chinese population. We could highlight, for example, the pathogenic strain of the German outbreak, and they are very different, and they can be distinguished pretty much. Take this approach to the general case, and you have Strainflan, which is basically typing phylogenetically at strain level using the MetaFlan2 markers. And this has been the work of Duitin Truong in our lab. Or take it from the genomics point of view again, and this is the work of Matthias Scholz, which is Pamflan, and this is a kind of different approach. We are typing the strain basing on the presence in red or the absence in yellow of specific strain-specific genes. So in red here you see the pangenome. Again, this is the dataset, the same dataset as before, E. coli. And we can see pretty much the presence, the specific presence of some strain-specific features of the German outbreak strains. And if we look at what features these are, they are the Shiga toxin and the antimicrobial resistance, these factors that we already knew that were of the essence in defining the outbreak, the strain of the outbreak. We could point out at the biogeography of Eubacterium rectale with strain-flan. This is done. Again, they are colored by nationality, and you can see this blue blob here in the graph, in the PCA graph, and again here in phylogenetic space. And these are all people from the Chinese population, so this is pretty much interesting. But again, this is on the human microbiome side. And the question we want to ask is, can we do the same with CITIC? Can we do the same with the urban microbiome, which is characterized by more complexity, less reference genomes available, whatever is there is less known than pathogens already known in the human gut or human skin. So it's an unexplored country, and we may want to test our tools, which are already available, already well tested on the human microbiome side, but do these tools work on the urban microbiome? And I'm showing you some example of this applied to the MetaSUB CAMDA dataset. We analyzed all the 1,600 samples from the New York, Boston, and Sacramento subway. And we could see that there is quite a lot of diversity there. This is the MetaFlan2 map, the top 70 species, and on here, and these are the samples on the rows. And we can see that there are bacteria that are more abundant, more prevalent across the same in the same environment. And for example, here we see that Pseudomonas tutseri. But again, we may ask ourselves, is this the same Pseudomonas tutseri? Which are the differences in this Pseudomonas tutseri? Is this the same? Is this the same person going around the metro and bringing it around, or are these different? And how are they different? And this is kind of the central point of why we need to go to strain level to uncover this diversity and to answer this question. So an example is Metamlesti applied to the entire dataset of New York, and we could see the presence of well-known both commensal and opportunistic pathogen bacteria. And again, these are the species, these are the Metamlesti counts of total and known and new strains for each species. And we see some known bacteria like Acinetobacter baumannii, Enterobacter cloacae, and Stereotyphomonas Mytophilia. And some of them are new, so they are observed here for the first time. They've never been, these combinations of the typing loci used in the approach were never observed before. And taking a closer look to Stereotyphomonas Mytophilia, which is one of the most interesting opportunistic pathogens if you're immunocompromised, one of the most dangerous, we could see that both new and known strains were detectable, not in one, in some cases, not in one case, but in multiple cases, sometimes on the same train, sometimes in the same station. And this is kind of expected, because if you sample the same train or the same ticketing machine twice, you would expect to see the same strain there. So that's quite expected. But we can have this kind of resolution even in urban microbiomes, and that wasn't given. Wasn't a given thing. We could highlight the diversity of E. coli, pretty much as I've shown before, again in the New York subway. The yellow spots are the subway reconstructed strains, the others one are from literature. And we could highlight the presence of different phylotypes in E. coli in the New York subway. There's the phylotype A, there are other phylotypes commonly associated with the human gut. And this is pretty much interesting to be able to reconstruct this kind of information from there. But we may ask ourselves, what is the strain landscape? How many strains are per species and per sample? And we could do that because we could reconstruct the genome of the strains, of the individual strains with strain-flan. And looking at the sequences, we could ask for each nucleotide, for each base, what is the information that supports the idea of more than one strain in that sample. And this is what we're looking here. So each point is a strain, hence is a sample. And we are plotting the non-polymorphic sites. So how many sites in the sequence do not support the idea of multi-strains. So the higher the point, the higher on the y-axis, the more the sample is prone to have a single strain in it. The lower is the coordinate, the lower is the value, then the more the sample is bearing more than one strain. This is the supporting information. And we see different behavior, species, this is by species. For example, A. radioresistance had a more dominant landscape, whereas P. acnes had more. The samples were highlighting a more multi-strain hypothesis. And this is pretty much expected if you think that P. acnes is brought around by people. It's essentially a skin microbe, so many people touching the same surfaces, and it might be expected that there are more strains on the same surfaces. And in between, for example, Pseudomonas tuceri, the red bar here, which shows a different mixed approach. Some samples are dominated by a single strain, some samples are not, are dominated by multi-strains. And again, this is very interesting to be observed from metagenomics. And we had some similar results in the human microbiome applying the same tools, but we had never observed these kind of features into urban and built environment microbiomes. We could go a step further and look at Pseudomonas tuceri, which is the most abundant in the New York subway. That's why I'm showing you this one in PCA space. So we reconstructed the sequence of the strain and plotted it in PCA space. And each point here is a sample, and they are colored by New York's wards. And they do not appear to correlate very much with New York wards, although there appear to be 2 or even 3 clusters of Pseudomonas tuceri. So the number of strains that are reconstructible in New York subways of Pseudomonas tuceri are essentially 3 in this case. But we could do a step further again and ask ourselves, how are these strains different? What are the features that characterize these strains? And one of the most interesting questions we may ask is about antibiotic resistance. And so we may ask Pamphlan to look at the differences at the different strain and selecting those of them that are positive to a given antimicrobial resistant gene. And that's what we did here with the MEX-A, which is a part of the a multi-drug efflux pump responsible for resistance to multiple antibiotics, such as chloramphenicol. And we may see that some strains, although the strains on the left is more prone to encode for this kind of multi-drug efflux pump, there were also other elements on the other side, on this strain, which some of them have the gene. And we may think that these are examples of horizontal gene transfer, which are totally possible in the case of especially Pseudomonas, which are extremely plastic genomes. But this is really interesting because we may make a little step further even in the definition of strains. So these are 2 strains, but some members of a particular strain are encoding for the antimicrobial resistant genes, antibiotic resistant genes that we are interested in, and that's terrific. We could do the same with Stereotyphomonas Mytophilia. Again, we did the same thing here, and there appears to be 3 different clusters, 3 different strains, if you may, of Stereotyphomonas Mytophilia. And this is really interesting because if you think of the diversity in the microbiome, of the different combinations that the genetic features could appear in, we see that there is essentially a limited set of genetic features that specific strains may have in an environment so big as the New York subway. We could do the same into Boston subway, and we could profile the Propionibacterium acnes, which was again the most abundant species, and we could do phylogenetic analysis. And although the data did not correlate with lines and trains, we could see some specific cases in which the same or closely related strain of P. acnes was recoverable from the same line or from the same train. So this is again an example of how we can track the strain into the different environments of the subway. And we could do the same with viruses, which are extremely interesting, and this is a quite recent line of research we have established in our lab. And we could do the same kind of reconstruction basing on in this case, the full genomes of viruses from the New York subway again. And although I don't have time to go through this in detail, I wanted to give you this brief example of the bacillus phages. We could detect the lactobacilli in the New York subway, and we could also detect the lactobacillus, the bacillus phages, and profile them phylogenetically and reconstruct their entire genomes, and be able to do this very nice analysis in phylogenetics of the viruses. And we are currently profiling the whole virome of the New York subway. This is an ongoing work. So I don't have time to go through this in detail, but I wish also to invite you to my poster, which is specifically devoted to the viral diversity into metagenomes, including environmental metagenomes. We have a very nice work ongoing on the alpine lakes metagenomes, and especially devoted to viruses. I will be there on Tuesday, and the poster is the B60. So to conclude, I hope I have convinced you that going to strain level is of the essence when looking at complex communities. And we've already been doing this for quite a long time on human microbiomes, but the entire settings of cities and urban and built environment is rather new. And we wanted to test if these tools are working on this kind of more complex and probably less known data set, and I think we did, and we achieved strain level resolution in urban microbiomes, specifically for bacteria, but we have some insights also onto micro-eukaryotes, which are very interesting as well. Of course, much work has still to be done, and I'm talking about the so-called dark matter, which is the whatever is still transparent to our pipelines. And I'm sure all of you already heard the term. And this is an extremely difficult and extremely daunting task, but it's also very exciting because no one has ever observed this kind of variation, this kind of microbes, species that no one may even think of discovering. And if I may, probably in built environments, in environmental metagenomics in general, resides a huge variation, a huge species and strain-specific variation that we still don't know nothing of, and that's thrilling. That's really interesting. So, to conclude, I wish to thank all my wonderful colleagues at the Laboratory of Computational Metagenomics, all the people who took part into these studies, and again, the organizers for the opportunity to speak here today, and last but not least, you for your kind attention. Thank you very much indeed.
Great talk. Any questions?
Hi, this was excellent, and looking at Chris now, I'm thinking maybe this was already suggested, but as a comment, you should have really just taken a subway map and placed things on there. I'm sure that would explain lots of variation that you're observing. So just as a comment. From the strain resolution story, why do you believe, let's put it another way, I don't believe it, that strain resolution is going to be the answer to this. I think that you need to go much, much more down, particularly when dealing with 99.9% of the bacteria, which you don't have cultured and you will never have cultured. So you need to go more than strain resolution down. You need to go to function level and maybe look at functional clusters rather than particular bacterial signatures. Have you thought about this? Have you considered that?
Well, yes, and indeed this is kind of the type of analysis we've been doing with the specific case of the antimicrobial resistant gene, this is the question. This is a very specific question we may want to ask: is what is the proportion of strains resistant to that antibiotic and not? But looking at the functional potential is surely important, and that's a complementary part of our work, I think. And coupling together the genetic variation and the functional variation is crucial. What we see in the human microbiomes, for example, is that sometimes if you focus on the functional variations, you don't see much of the difference that you would see with strain level resolution. And that's because sometimes the same pathway, the same function is performed by different bacteria. So if there is a variation, you don't see it by looking at the specific pathway. And this is why the two things should go in parallel, should be.
So I guess I'm just pointing out that particular strain diversity, or whatever you want to call it, whatever level you go down beyond strain, right? Maybe individual cell microbial diversity is not necessarily the thing that drives the particular observed phenotype of, okay, human, but also subway, I don't know, soil. So if you took the same sample, this is the worst case scenario, you took the same sample, you submitted it to two different analysis pipelines or two different databases, they will get very different strains that are there. Do you care? Do you only care about the functions that these things do? Or do you actually care? Okay, this is Alex and this is Peter and whatever other name you come up with for the strain.
I think you care of the information. Again, functional information is important, but it's not everything. And if two strains are different, let's talk for the sake of simplicity of the same species. And we see two different strains of E. coli encoding for the same toxin, for example. Mm-hmm. And this is pretty much the point. We have two different populations of E. coli, which happen to encode for the same protein, but we don't know why, and we may we can discriminate between them because they are different at other levels in their genome. But if we only focus on the toxin, those two strains may appear equal to our pipelines, and that's not maybe the case. Maybe, I don't know, I'm just running through, but I don't know, maybe one of them has a particular antibiotic resistance coupled with the toxin, the other one doesn't. And you may need this kind of information. Again, this is functional. You can discover this by functional analysis as well. But the point is that there is much more than function in different bacterial populations. Strains or below strains is important, whether you think they are specific genetic features or genomic features. And again, you go to functional again. So the line is a bit blunt, I guess.
Can we talk about this?
Sure. Maybe I didn't understand the question correctly. That's
okay.
Well, yeah, I think we all want both of those things all the time. And just as a curiosity to follow up, if you run, say, Human, which I know is in the same MetaFlan and Flan tools on some of these kind of samples as well.
Yeah. Well, Human can be applied in principle. It can be applied to these kind of samples. We didn't do for the specific challenge, but we wanted to look at genetic primarily, but in principle this should work.
Yeah. One other question?
Thank you very much. I have a question concerning very clonal bacterial species. So how do you deal or how do you calibrate your data set when you would look, for example, at bacillus and traces or mycobacteria? They don't show the same variability like E. coli, for example, but somehow minute differences are there, much more important than they are in E. coli. So yes. Have you some system, you showed the bacteria, some of them, they show a big diversity, and your conclusion was, okay, there are many people having different strains, but maybe in mycobacteria you wouldn't see this on this level. And there are many people from different countries also bringing the microbacteria, but you wouldn't see, maybe if you don't have a little bit more diversity in your tools, you wouldn't see the differences at all. Do you have some ideas there?
Yes, I think that depends on how fine you set the strainer and how fine how many. For example, I'm taking the example of Metamlesti, for example, because I personally developed it. And the way the protocol works, so even in molecular biology settings, is that even one SNP of difference between the candidate, the selected locus, is enough to classify for a new species. And there is a debate on whether we should care of 1 SNP, 5 SNP, or 10 SNPs, and that's probably even species specific, because there are 5 SNP of variation in Pseudomonas are a thing, and in Eubacterium are another. But at the level we set the analysis here, even with E. coli, we were looking at strains with at most 1 SNP of difference. So if even 1 SNP was enough. And you may think that these are random variations or just not enough to call for a new strain. That's why you couple different tools together to have the widest picture possible. So I don't believe there are 2 strains only based on Metamlesti analysis, but when all these different approaches are kind of telling me the same thing, then that's quite. It's not really strain in biological, in a molecular biology sense, but we can discriminate population based on genetics, and that's pretty much the point.
Great. I want to thank you very much for the great talk. Thanks.